Hi, I'm Banu and I will present TargetCall, which is analyzing these nanopore signals in a different way. The title of our work is Eliminating the Wasted Computation-Based Calling via Pre-Based Calling Filtering. And our summary is, so we realize that base calling can consume up to 84.2% of the total execution time in entire genome analysis pipeline, and it becomes a bottleneck for this pipeline. But what's the real problem is that majority of this costly computation is wasted, as majority of the reads do not match the reference that's of interest by the application, and they are wasting the base calling computation. So what we aim in this work is to have a method that would eliminate this wasted computation base calling while maintaining high accuracy, scalability, and adaptability. Our idea is to have a pre-base calling filter that would filter out the user's read before base calling with a highly accurate filter, with high accuracy and high performance. For this, we propose TargetCall, which is a new pre-base calling filter that works in two steps. First, we have a lightweight base caller, which we call as light call, that performs base calling in a noisy way compared to state-of-the-art base callers. And then second, there's a similarity check component that compares these noise reads to the reference genome to determine the similarity of these reads and kind of find out if the read is useless or useful. Our results shows that with TargetCall, we can improve the end-to-end performance of base calling by 3.3x compared to state-of-the-art base callers by filtering out majority of the useless reads. And we show that if you were to repurpose the adaptive sampling methods that John presented or discussed as pre-base calling filters, TargetCall achieves better performance, throughput, recall, and precision than them. And we open source our work in our GitHub page. I will start with background, but John already gave a lot of the background, so I will skip these parts. As John said, genome sequencing is important in many areas, and nanopore sequencing is a widely used sequencing technology. It has long reads, high throughput, and low cost. A traditional nanopore sequencing pipeline starts with genome sequencing, where the reads are passing through a nanopore, and as this is happening, nanopore sequencers are generating a raw signal. This raw signal then base called using complex neural network models that would convert these raw signals to a set of bases. And the next step after this is to perform read mapping, where these reads are matched to the reference genome to compute if the read can be mapped or not mapped to the reference genome. And prior studies shows that only the base calling step can consume up to 84.2% of the total execution time, while the remaining analysis takes less than 16%. That's why we conclude that base calling is really a major computational overhead in nanopore sequence analysis pipeline. But our real observation in this work is that majority of these base call reads are discarded after base calling in the later steps of the downstream analysis. So one example for this applications is the SARS-CoV-2 genome assembly, where only less than 1% of the reads are useful, while the entire read set is processed. And typically the reason why 99% of the reads are used is that they do not match the reference genome that is targeted by the application, which is SARS-CoV-2 reference genome in this use case. That's why we say that users reads waste significant amount of base calling computation in traditional pipeline. One option is targeted sequencing or adaptive sampling, as John presented, which is in contrast to the traditional pipeline where we sequence all reads and base call all of them. In targeted sequencing, what we do is we selectively sequence only the useful reads. And for this, we add a step in the sequencing part where we identify if the read is going to be useful, and then if it's not, the nanopore sequencers can stop the sequencing of the user's reads by rejecting them from the pore. But this requires a method, and as you can see, the rejected ones are stopped and the accepted ones are fully sequenced and then sent to the base calling step. But this requires a method to identify which reads are useful and which reads are going to be useless. And some of these methods are presented by John, such as uncalled, SIGMAP, and RowHammer. But we identified several limitations of these prior tools, and we state that like all of these approaches at least suffer from one of the three following key shortcomings. First, they have low sensitivity and they falsely reject many useful reads. Or they have poor scalability in the sense that their performance and accuracy drop significantly for large reference genomes. Or they have lack of adaptability, which means that for each kind of genome sequencing application that you are targeting, or each kind of different reference genomes that you're targeting, you need to retrain neural networks, which is a costly method to do. That's why we say that these targeted sequencing methods, although they are useful for real-time analysis, they are not robust approaches for eliminating the wasted computation in base calling. What we have as our goal in this work is to eliminate the wasted computation in base calling, but while maintaining high sensitivity in keeping useful reads, and while being scalable to large reference genome, and while maintaining adaptability to different use cases without requiring any further preprocessing steps. Next, we present TargetCall, which is our pre-base calling filter. If you remember our approaches, our first option was to have the traditional pipeline where we sequenced and base call all reads. And our second option was to do targeted sequencing, where we base call everything that is sequenced, but we selected the sequence things depending on if they're going to be useful. There's like a prediction step. And then we only do sequence the useful ones, or the ones that are predicted as useful, and then we base call everything that's coming from the sequencing step. What we propose instead is to have a pre-base calling filter, which is to selectively base call the useful reads. So in contrast to the traditional pipeline, we sequence everything similar to the traditional pipeline, but we don't base call everything. And in contrast to the targeted sequencing, we sequence everything, but the targeted sequencing does not sequence everything. But the thing is, this requires a method or like another step in the pipeline, so pre-base calling filter between the sequencing and base calling step. And this filter needs to identify whether the read is going to be useful or useless with high accuracy. And in TargetCall, anything that is identified as useless are discarded from the further analysis in the pipeline, and useful reads are forwarded to base calling step to maintain the base calling accuracy for the useful reads. So in high-level overview, TargetCall works in two steps, and then with a high-level goal of predicting if the read is going to be useful. And the first step of TargetCall is light call, which is a lightweight base caller, and it does base calling, but in a noisy way. Noisy in the sense that the accuracy of individual bases are not as high as the state-of-the-art base calling, but it's still high enough to compute if the read is going to be similar to the reference genome if you were to do the state-of-the-art base callers. And after light call computes the noise reads, there's a second step, which is the similarity check component that compares these noise reads to the reference genome to predict if the read is going to be useful. And as I said before, anything that is useless is discarded from further analysis in TargetCall. Anything that is useful is forwarded to the base caller so that we maintain the base calling accuracy for the useful reads. In contrast to the prior methods, TargetCall has high sensitivity because we have a highly accurate similarity check component. It has good scalability because the light calls model's performance is independent of the reference genome size, and it's adaptable to different use cases without requiring any other processing step or any other neural network training, because the light call model is generic. That means like you just use the same light call model regardless of your read set and the reference that you are using. And that's why TargetCall is a robust solution for eliminating the wasted computation and base calling. Now we can explain our two steps in a bit more detail. And the first step is light call, which is a lightweight base caller that produces noise reads compared to the state-of-the-art base callers. And light call is designed based on simplifying state-of-the-art base caller model architectures while maintaining most of the accuracy benefits, but reducing the execution time significantly. And we made this simplification in 3 steps mainly. First, we reduce the channel size of the convolution layers. Second, we remove the skip connections. And third, we reduce the number of basic convolutional blocks in the state-of-the-art neural network models for base calling. And the final light call architecture is composed of repeated convolutional blocks, where each block has one or two components that include group convolution, pointwise convolution, batch normalization, and read layers. And the second component is similarity check, which has input of a noise read coming from LightCall and the reference genome that is determined based on the application that we are targeting. And the goal is to find if these two things are similar. And depending on the answer, the read is going to be useful or useless. So in some applications, the read is going to be useful if it's similar, and in some other applications, the read is going to be useful if it's not similar to the reference genome. But for this model, we didn't design anything new. We just used the state-of-the-art read mapper, Minimap 2. And we showed that in the paper while using LightCall and similarity check together. So similarity check or Minimap 2 using the noise reads coming from LightCall, we can achieve up to 99.45% sensitivity in keeping useful reads. So that shows that like the minor or the noise reads coming from LightCall is not noisy enough to hinder our ability to find if the read is going to be useful. And we conclude that and show that in the paper that 0.55% of the loss of the useful reads during the filtering time can be tolerated by sequencing depth of coverage that is already required for accurate genome sequence analysis. And we evaluate TargetCall using 3 different use cases. We choose 3 different use cases in a way that we show how scalable and adaptable TargetCall is. Our first use case is SARS-CoV-2 detection, where we have a small COVID reference genome. And our sample of reads contains both COVID reads and human reads. And our goal is to identify COVID reads among this sample. So we want to eliminate the human ones. And if the read is now in this use case similar to the reference genome, which is the COVID reference genome, the read is identified as useful because we are targeting COVID references, and otherwise the read is identified as useless. Viral detection is also a similar use case, but this time the difference is our reference genome is not one single viral reference genome, but it's composed of multiple of them. So we have a bit more complex reference genome, and our sample now contains bacterial and viral reads, and we want to identify the viral ones. Here now, the viral and bacterial reads are resembling each other more compared to human ones. That's why it's a bit like a different use case. But the approach is similar, and anything that is similar to the reference genome is identified as useful for this use case as well. What's different is the sepsis detection use case, where we have a large human reference genome. And this is to show that like TargetCall can scale well to the large reference genomes, and we have a human reference. And what we want to identify is all the bacterial reads in a sample that has bacterial and human reads. The difference is this time we don't know which bacterial reads we are looking for. Basically, we are checking for any kind of bacterial read in our sample. That's why instead of searching for specific ones, we just filter out what we don't want, which is the human reads. And that's why in this use case, anything that is resembling the reference genome is identified as useless, unlike the other ones. For our evaluations, we have 2 sets of experiments. First, to show the benefits of pre-base calling filtering, where we have the baseline as Bonito, which is a state-of-the-art base caller, and we compare the Bonito alone and Bonito preceded by TargetCall to perform pre-base calling filtering. And our evaluation metric is base calling speed up, at least for this presentation, and you can find a bit more results in the paper. And the second set of experiment is to compare TargetCall against targeted sequencing approaches. And we also compared it with OnCall and SIGMAP because these are the ones that are fully open source and also can be applicable as pre-base calling filters without any other changes. And our methodology is to repurpose these methods so these methods also require a labeling mechanism to predict if the read is going to be useful and then rejecting the user's reads. We just repurpose these labeling mechanisms to perform pre-base calling filtering instead of adaptive sampling. Basically, we just compare TargetCall, Bonito pressed by TargetCall, OnCall, and SIGMAP in terms of their execution time, recall, and precision, and more in the paper for throughput. We have various data sets, 5 different resets from various organisms, most of them sampled from prior work, and then one we simulated using Deep Simulator because we couldn't find any proper real data set for this type of viral reference mix. And we open source all our data sets so that you can reproduce our results. We have 4 different reference genomes with various reference genome sizes and ratio of useful reads. to show that like we really can do very well in different kind of scenarios. So we evaluated TargetCall using 2 GPUs, NVIDIA 100 and the Titan V for light call, an AMD system with 0.2 terabyte main memory for the similarity check component. But for evaluating SigmaP and uncalled, we use a similar AMD system with 1 terabyte main memory because SigmaP and uncalled require more memory than we have in the first system for large reference genomes. Our first metric is the base calling speedup that we can achieve with TargetCall. So as you can see in the y-axis, we have the execution time, and in the x-axis, we have different use cases as well as the average. So this blue bar shows the execution time of Bonito for each use case and the average. And then the green bar shows the TargetCall execution time plus the Bonito execution time for reads that pass the TargetCall filter. So it's really an end-to-end comparison of the base calling times. And we conclude that on average, we can get a 3.3x speed up, base calling speed up over Bonito. And show that TargetCall is really beneficial for improving the base calling execution time. And when we compare TargetCall against state-of-the-art approaches in terms of its performance, again, all bars include both the execution time of each specific tool and the base calling time of the reads that pass by these individual filters. Our first conclusion is that TargetCall can provide up to 9.7x speed up over prior targeted sequencing approaches. And more specifically, the speed up improvement is speed up improvement is larger for larger reference genomes, more specifically up to 13.3x over SigmaP, and we were even unable to compare it against uncalled because uncalled couldn't generate the index for such a large reference genomes. And next, we compared the recall of these tools, and recall is referring to the ratio of keeping useful reads among all the useful reads. And on average, TargetCall's recall is 99.1%, which is quite high. And it's actually quite better than prior approaches, up to 23.2% better recall over SigmaP. And similar to the performance benefits of TargetCall, recall benefits of TargetCall also improve with increasing reference genome size, where we can provide up to 15% better recall compared to prior tools on average. So there's one caveat in this plot, which is that in sepsis use case where we have a large reference genome, we don't actually measure the recall for finding human reads, because here the useful reads are the bacterial reads. So if the human reads is not matched to the human reference genome by this use case, they just pass the filter in a wrong way, and it's not a problem for the bacterial reads that are the real useful reads. But what we wanted to test is what if we had a use case where the goal is to keep the human reads in a sample, and we have a human reference genome as well, and then we find that like the recall for finding human reads for SigmaP and uncalled is quite low. It's like on average is like 47% or something. And this is recall for finding human reads in a human sample, whereas TargetCall provided more than 96% recall for the same scenario. That's why we conclude that TargetCall consistently provides high recall for all reference genome sizes tested, regardless of the type of experiment that we designed. And lastly, we compare the precision of these TargetCall against these adaptive sampling approaches. First, we concluded TargetCall's precision is on average more than 92%. Which shows that TargetCall can really eliminate the wasted computation in base calling by filtering out the user's reads before the base calling step. And on average, our second conclusion is that on average, TargetCall can provide up to 60% better precision than prior tools. We have more details in the paper about like details of how these targeted sequencing methods works individually, and then what are their limitations, and what is our light call design in a more specific way, and why we chose to do a specific type of optimizations while improving the performance of the light call. and then details on our evaluation methodology and more detailed results on different target call designs, many different light call architectures that we used for target call design to see the base calling speed up, recall, and precision of these different light call architectures to finalize our design, and end-to-end accuracy and performance improvement of target call on entire genome analysis pipeline, including the variant calling step, and throughput comparison of target call against our SigmaPen uncall, where we show sometimes more than 1100x better throughput than prior tools, which really shows that target call can be applied for real-time analysis as well. And a peak memory discussion between these tools and then target call, which shows that target calls like peak memory requirement is much less than prior tools. Please check out the paper for more details about our results and everything else that we discussed. And check our GitHub page if you would like to test target call and verify our results or reproduce our results. And let me conclude with that. So target call is an accurate, scalable, and adaptable pre-base calling filter that has two-step approach. First, a lightweight base caller, light call that does noisy base calling with high performance, and second, similarity check component to compute if the read or predict if the read is going to be useful for the later downstream analysis by computing the similarity of the noisy read coming from light call to the reference genome that is identified based on the use case. And our evaluations of target call shows that we can improve the base calling performance significantly for all use cases that we tested by filtering out majority of the users' reads. And we also show that target call has better recall, precision, performance, and throughput than these adaptive sampling approaches if you were to repurpose them as pre-base calling filters.